cell injury. Chronic inflammation, remember we said active inflammation, the tissue injury as well as the repair occurs at the same time. We spoke about the word fibrosis a lot as well. So today we're going to be talking all about it. So tissue repair, okay, just repair. Anything you have and you want to get it repaired, what is the main purpose behind you getting it repaired? Yes?
To make
Restore the function, yes, what else?
it function properly.
Yeah, make it function properly, restore its function, what else? Yeah, there is something that has gone wrong, you were not trying to get it fixed. Structure. So your car gets an accident, you go to the gentleman, what do you tell him? You tell him I want it to work just like it was before and look exactly the way it was before the accident, isn't it? So whenever we repair to make sure that that thing's structure and function is restored. So tissue repair involves restoration of the tissue architecture, the structure and the function after an injury. Now this repair can be done by two processes. One is the regeneration, which involves proliferation of residual undamaged cells. So wherever there is an injury, there will be cells in the surrounding which are going to be normal, they will not be damaged. So regeneration involves the proliferation of those cells, they divide and fill the gap where the injury had occurred. But this regeneration would occur in dividing or non-dividing cells. Dividing cells only. Can non-dividing cells proliferate? No, they don't. For them, which are the permanent cells of the non-dividing cells, the injured tissue which is incapable of complete restitution and regeneration, in that case you have the scar formation. So remember one thing. Healing or repair by connective tissue, repair by scar formation, fibrosis, this is all the same thing. So regeneration will occur only in the, and this is a very important thing I'm saying now, regeneration will occur only in the cells which have the ability to divide. Whereas scar formation will occur in the cells which do not have the ability to divide, as well as the dividing cells when their extracellular matrix is destroyed or damaged. Cells, they divide, they have the ability to divide, but they will only divide when they get the growth factors from the extracellular matrix. They have the ability to divide, but extracellular matrix is damaged, there is no growth factor available. Will they divide? No, they have the ability, but they cannot. Why? Because they don't have support from the extracellular matrix. So remember, regeneration is one way of tissue repair that occurs only in the cells which have the ability to divide. Whereas the scar formation, fibrosis, or repair by connective tissue occurs in the cells which do not have the ability to divide, which are the permanent cells, as well as in the dividing cells when their extracellular matrix, which means the supporting structure, is severely damaged. How will the scar formation occur? By the deposition of the collagen or the connective tissue. Main content is the collagen. Laying down of the connective tissue, the collagen. What cell do you think would be depositing the collagen? Muscles? No. Fibroblasts. They will be responsible for that. And hence we call the thing fibrosis, which is extensive deposition of collagen in the tissues when they undergo chronic inflammation. Skin, lung, liver, kidney. Do you think all these are dividing or non-dividing? Skin, liver, kidney.
Dividing.
They have the ability to divide. But what happens in chronic inflammation is that their extracellular matrix is destroyed, and therefore the repair of these tissue will occur by the process of fibrosis, which is the scar formation by the deposition of collagen. Also, in acute inflammation, whenever you have purulent localized inflammation in the form of abscess, it is going to heal by fibrosis, which is deposition of collagen. Any extensive damage to the tissue during acute inflammation will also repair by fibrosis. You remember in chronic inflammation you have the granulomas, the fibroblasts surrounding it. So how will the granuloma repair or heal? By fibrosis, deposition of collagen in that tissue. Now this is just to tell you your cell cycle. G1 phase, cell increases its contents. S phase, it duplicates its chromosomal number from 46 to 92. G2 phase, it looks at any errors during this replication of chromosomes. And the M phase is when the cell divides into two cells containing 46 chromosomes each. This is the normal cell cycle, isn't it? There are some tissues called labile tissues. They are always dividing. Their cycle is continuously going on. We will discuss them in detail in coming slides. Then you have stable tissues. They are called quiescent tissues, quiet, from quiet. They divide. After one division, they enter into a G0 phase, which is a quiet phase, quiescent phase. They go into sleep mode. They have the ability to divide, but now they are not dividing. When will they divide? When there is a stimulus, when there is an injury. It's just like, you sleep your computer, it sleeps. The screen is black. You just have to put an enter, it comes back again, isn't it? So they go into sleep mode. As soon as there is an injury to that tissue, they enter back into the cell cycle and start dividing again. So they are the stable tissues. And then you have the permanent tissues, which before the birth of the baby, they are dividing, but once the baby is born, they become permanent cells with their own special structure and function. They will never be able to divide. And the differentiation means that the process through which the cell becomes specific with regards to its structure and function. Now the cell has a specific structure, it will always remain the same, and has a specific function, which will always remain the same. So this is your cell differentiation. Let's discuss these tissues which we said based upon their ability to proliferate. So labile tissues are those which are continuously dividing. Why they are continuously dividing? Because there are old mature cells which are being dying. They are continuously being lost. Skin cells, epithelial cells. Continuously the old cells are dying. And if the old cells die and we have no more new cells, that organ or tissue will finish, isn't it? So continuously the skin epithelial cells, the epithelium, anywhere else in the body, we'll discuss that. All those, the old cells are dying, the new ones are taking their place. How that happens? Number one is by maturation of stem cells. We have stem cells at the basal layer. They divide, increase their number, and they differentiate into that specific cell which are dying. Secondly, those specific cells which are not dead, they start to divide. So these are the two ways that by the maturation of stem cells as well as by proliferation of mature cells. So where are the labile tissues? What are the examples of labile tissue? Number one, hematopoietic cells of the bone marrow. So all the cells that are produced by the bone marrow, they have a special lifespan. For example, what is the lifespan of a red blood cell? 120 days. After 120 days, if today a red cell is produced by bone marrow, after 120 days it will die, isn't it? So if at 120 days all red cells are dying and the bone marrow does not produce new cells, we will have anemia and finally will die. So continuously, and it's not that all red blood cells were produced on day one, so all red blood cells finish at 120 days and then the new ones come. No. Every day some red blood cells are dying, it is their day 120, some new ones are being produced. Every day there are new cells which are produced to replace the ones which become dead. So this is a continuous division of the hematopoietic cells from the bone marrow. Then your surface epithelium. Wherever we have epithelium in the skin, we have epithelium in the oral cavity, in the mouth, we have epithelium in the GI tract, in the female genital tract, the cervix, the vagina, the uterus, the fallopian tubes, exocrine glands, urinary tract. All these are the examples of the organs which are lined by epithelium. And in the epithelium, as I told you in the aging, cells, they divide up to a certain number of cell divisions and then they stop dividing and then they die with apoptosis. So the old cells, which are exhausted cells, they are dying, they are shed off, and the new cells take their place, and these tissues are called the labile. Stable tissues, as I told you, they are quiescent. They are in the G0 stage of the cell cycle. Yes, they have the ability to divide, but they are not dividing. When they are in the G0 stage, they have minimal replicative activity. Either they don't divide at all, or if they are dividing, it's so less it does not make any difference at all. But because they have the ability to divide, when will they start dividing? Whenever there is an injury to that tissue. Automatically they will start dividing, and one of the examples of that is your liver. Liver is exceptional in stable tissues. Stable tissues, yes, they have the ability to divide or regenerate, but not as much as the labile tissues. But in the stable tissue, there is one organ which has a very high replicative ability, and that is the liver. So if I ask you, which one has more proliferative capacity, the labile tissue or the stable tissue?
Labile.
Labile. And in the stable tissue, which one has the highest ability to divide?
Liver.
The liver. Very good. So is our liver cells dividing right now? If they would divide, then our liver will be that big, it will come out of our body. It has made the organ, if the organ's finished, the cells are in G0 phase, quiescent phase. Now, if I give a part of my liver as a donation to one of my loved ones for a transplant, that the liver cell starts to proliferate again, and until they make my liver back to where it was. Similarly, a person has a tumor, you can take away the part of the liver, the tumor one with a tumor, the remaining liver will start dividing, it will make the liver back again. Then, what are the other examples of the stable tissue? All solid organs. Your pancreas, lungs, your liver, kidney, spleen, all these are the examples of the stable tissue because they are the organs which have the ability to divide, but they will only divide when there is going to be an injury. Endothelial cells, what do the endothelial cells make? Blood vessels, very good. So are the endothelial cells in our body dividing all the time? If they would have been dividing all the time, we will have blood vessels coming out of our body. Big blood vessels because they're continuously dividing. No, they are not labile tissue. Labile tissue continuously divide. They will only divide when needed. So when will endothelial cells divide? When there is tissue repair. Angiogenesis, new blood vessel formation. Remember that? I told you whenever there is repair, we need good blood supply. So repaired tissue will make new blood vessels. I will tell you how it happens in today's lecture. So, or in the lecture tomorrow actually. So, endothelial cells, they are also stable cells, will only divide when there is an injury and we need to repair that injury. Fibroblasts, what is their function? Yeah, to produce what? Collagen, which is involved in fibrosis, of course. So, are they producing collagen all the time? No, they will only proliferate, divide at the site of injury where we want to repair by scar formation, that they will be proliferating, increasing their number to produce more collagen. Similarly, the smooth muscle cells in the uterus, let's say, when they have a stimulus in the form of estrogen, they divide. Once the estrogen levels go away and progesterone takes over, they stop dividing. Then they undergo apoptosis when the menstruation occurs. So this is also an example of your stable tissue which has the ability to divide, only will divide in response to a stimulus, which most of the time is the injury. Permanent tissues, terminally differentiated. After one cycle, they become mature with regard to their structure and function. They don't have any proliferative capacity in the postnatal life after the birth of the baby. Neural tissue, your brain, spinal cord, the nerves, cardiac muscle cells, skeletal muscle cells, all these are examples of the permanent tissue. Whenever they will be damaged, how will they repair? Regeneration or scar formation? Scar formation, why? Because they cannot divide. But even those who can divide, can there be scar formation there? Yes. What's the condition? Damage, exactly. In order to make sure that the regeneration is involved in the repair fully, we need to have an intact extracellular matrix. Yes?
Now when time is the development phase with organs closed, they're important organs to the heart, etc.
Perfect.
Those usually have permanent
Yeah, they're also developing in the prenatal life before the baby is born, and it is developing. The baby is growing. The brain is also growing. Skeleton. So they grow within that. But once they are born, once they are born, they will not increase in the number, but they can increase in the size.
But the cell count is the same from birth.
Exactly. The cells are going to be exactly the same. That's why your brain, when you are a baby, and when you are really grown up, there's not a very big difference in the size of the brain. Okay. It doesn't happen that the baby has a brain like this and the person, no. There's not a big difference in those. Okay? And the skeletal muscles also, when they increase, when the baby is using it, the bones are growing, the muscles are attached to the bones, they are stretched, they increase in their size with the hypertrophy, but they do not increase in their numbers. The numbers are going to be the same.
The child gets damage to their heart, let's say within pregnancy.
Yes, that's how it will stay forever. Yeah, we cannot really fix it. That's the problem with the permanent tissue. See, this is again the same thing. Labile cells, like your epithelium, GI tract, whatever, always dividing. Stable tissues, like the liver, enter into the G0 phase. Will stay there, quiet. until there is an injury that they enter back into the cell cycle, start dividing, producing new cells. Permanent cells, when they divide, they become permanent. In the postnatal life, they are not going to divide at all. So these are like neurons and the cardiac muscles. Now, role of the stem cells, very important. Stem cells are very, very important in the process of repair. So in the labile tissues, the mature cells are short-lived. They die after a certain time, and the new cells take their place. How? By the differentiation of the stem cells. So there is always an equilibrium between those who are dying and those which are replicating to fulfill their number. So if more cells die and less stem cells are differentiated into that specific cell, what will happen to the tissue? The tissue will become thinner and thinner, isn't it? Because more cells are dying, less cells are replacing them. And if less cells are dying and more cells are being produced by the stem cells, then the tissue becomes too thick, isn't it? That's not what we want. That will affect the function. Both ways the function will be affected. So there has to be an equilibrium. As many cells die, approximately as many cells take their place, so the tissue remains the same in its structure and function. So this is very important. Where are the stem cells located? Always in the basal layer. At the layer in the bottom, over the connective tissue membrane. So in the epithelium, the skin, GI, wherever the epithelium is, they will lie right in the basal layer. They have two very important properties. Self-renewal capacity, they renew their numbers themselves. They never end. Tell me, if we have a lot of deaths of the epithelial cells and all the stem cells make the mature epithelial cells now to replace it, and then if they have another injury, they're going to say, oh sorry, we are finished with the stem cells. No. SubhanAllah, it's a creation of Allah, not a human being. You go to these big stores sometime, you ask them, I'm coming here for the fourth time, you don't have this thing. Every time I come there, my father needs a medicine, very normal one, I go to this big brand, Nahdi. Every time he says, we don't have it. This is what? This is bad management. If it is finished, they should bring it well in time that the customer comes, it is ready there. The demand and supply is the job of the manager to make sure things are there. We humans make such errors. It doesn't happen with the stem cells. That's sorry, we have to wait. Injured cells, bad cells, wait. I will give you new, we don't have stem cells now. No. They have self-renewal capacity. They have the stem cells, they divide themselves, increase their number. And then they have asymmetric replication. As the cells, they differentiate into the special type of cells, differentiation pathway, they make those cells which were dead. At the same time, they also retain their own renewal capacity. This is how it happens. Stem cells, they have a baseline population. Baseline population means at least they need this much population ready all the time. Clear? Like you say, okay, my goal is I should keep 10,000 riyals in my account all the time. I may need it for an emergency. See? Anything above, okay, I will spend wherever I want, whatever, but I should not get down. It's kind of a target, Some people could think like that. So the stem cells have a target that this should be their baseline population. So they will divide to produce this population. Now, suddenly, these were the cells here which have died after being matured, the epithelial cells. So these stem cells will mature or differentiate into these specific cells. So it's not that all of them differentiate into this and class we have no population of stem cells. No. They will keep dividing to keep this baseline population. In case their proliferation leads to lots of cells, more than what is the baseline population, they die with apoptosis. They maintain it. They don't want it to be too much. They don't want it to be less. And in case a lot of this is needed, then they proliferate very fast so that they keep this ready for a batch time anywhere if it's needed. Okay? This is called self-renewal capacity, and this is called the asymmetric replication. At the same time, they are differentiating into a special type of cells which are injured, and at the same time, they are also increasing their own numbers. This one, I just tell you the principle behind it. So I will not be able to answer too many questions because I am not a stem cell therapist or an immunologist, I'm a pathologist. But just to tell you about the stem cell therapy, how it occurs, because we're talking about stem cells. This is the embryonic stem cell, the embryonic oocyte. We removed its nucleus, so it's now eunucleated, isn't it? Now this is a cell, a permanent cell, or a cell from a person, which is a mature cell. We need it to produce large number of cells which are related to this cell. What we do, we take the nucleus out of this, we put that nucleus in this oocyte, which is the stem cell. Now this will divide, make a large number of cells. These cells will have nucleus of the adult cell, isn't it? Now nucleus has all the information about the proteins and which kind of proteins it will make, and so which kind of function it is going to do. So then they make these cells, like it can be, theoretically it can be muscles, the permanent cells, like the neurons, the heart muscle cells, and you can also make blood cells. So far, stem cell therapy has actually, in practice, helped in treating the patients who have bone marrow problems and who have cancers, blood cancers. There the stem cell therapy has really helped in producing the healthy cells using this technique. But when it comes to the real target of the permanent cells, because theoretically what we think is that, okay, if we put a cardiac muscle nucleus into the stem cells and we implant that stem cell in the area where there is myocardial infarction, it can divide and produce the myocardial cells, isn't it? This is theory. But they have failed to actually do it. There was lots of frauds related to this research. The governments did a lot of funding to the stem cell research, and unfortunately, they have not yet reached to anything to do with the nerve cells or the muscle cells. We just had a stem cell conference in Kmart about maybe 6 months ago, and it was very clearly stated there that the only where the stem cell therapy is working is the blood cells when it comes to treating the cancer and aplastic anemia and stuff like that. But for the permanent tissues, they have actually failed. But this is actually what I want you to tell in this slide is the principle behind the stem cell therapy is like this. You take out from the stem cell the nucleus, you put in the nucleus, and then it divides. Okay, now, growth factors are very important. That's the reason when I said that the cells which can regenerate, even they will have a healing or repair by scar formation if the extracellular matrix is damaged. Why extracellular matrix has the growth factors? Growth factors, they are responsible for stimulating the cell division. They are proteins and they bind to the receptor, isn't it? If there is no receptor, they will not work. But this is something very special. Same growth factor binds to a special receptor on one cell, cause it to divide, promote cell division. Same growth factor binds to another type of receptor on another cell, stops its cell division. So it's to do with specific receptors. There is a growth factor A. When it binds to the receptor B on one cell, it promotes its cell division. But the same growth factor A, when it binds to receptor C on another cell, it causes the cell division to stop. So the genes that control this proliferation are called proto-oncogenes. Just know their name. We will discuss about them in detail from next week when we start studying neoplasia. And growth factor can be specific to one cell, and it can be generalized to multiple cell types. For example, growth hormone. It affects all the cells in the body to grow, isn't it? But then you have a thyroid stimulating hormone. It only stimulates what? The thyroid cells, isn't it? So some of these growth factors, they are specific to a cell, and some have effect on multiple cells. Growth factors which are involved in our repair are mainly produced by the leukocytes, parenchymal cells, and the stromal cells, which are your extracellular matrix cells. Now, how do they work? Growth factors work through a signaling mechanisms called autocrine signaling, paracrine signaling, and endocrine signaling. Autocrine, see here, autopilot. The aeroplane is flying on its own. This cell does not depend on other cells. It produces its own growth factors. They bind to its own receptors and stimulate this cell to divide. Clear? Autocrine. Para, next to you, parallel to you. So paracrine signaling is that one cell produces the growth factor. They bind to the receptors on the adjacent cell, the cell next to it, and then it stimulates that cell to divide. And endocrine signaling, just like the endocrine hormones, these growth factors are produced by the cells. They are put in the blood. They travel in the blood to a greater distance, and far from where they are produced, they attach to the receptors of the cells and cause these cells to divide. So this is the endocrine signaling of the growth factors. So just some examples, those which promote the growth, the proliferation are epidermal growth factor, platelet-derived growth factor, fibroblast growth factor stimulates the fibroblast, interleukin-1, TNF, the master cytokines, they promote the fibrosis, vascular endothelial growth factor, you see the name, a growth factor that stimulates the endothelial cell proliferation of the blood vessels. So when we need new blood vessels, this is a very important growth factor. And those which stop the growth proliferation, you see, if the growth factors are all promoting, when they are released, it means that the cells will keep on growing. But once the liver is fully formed, we don't need it to be bigger now. Then those growth factors are neutralized, and those which are inhibitors, they take effect. That okay, now stop cell division. As I told you, I really don't believe how these people in the West are atheists. Even in our Muslim world, some very, very highly learned people, liberal people, they think that, Nauzubillah, there is no Allah subhanahu wa ta'ala. They're atheists. I don't know, especially being doctors, how do you think that this person in front of you is just made by an incident, or it's nature? Okay, this is just nature. I mean, there is no one who created it. So, okay, this is miserable, because this is just the knowledge which we have been able to extract from just this human being. That's why I say being doctors is much more responsibility than being engineers. You are working on a machine, it doesn't get fixed, you spoil it, you can take it to another friend of yours who may be more capable than you, and he'll help you fix it. But with a human being, you don't have a chance of error. And that's why the medical study is more extensive than the other studies, because you study the human being. And you see how these sensors work. Imagine, sometimes I sit in the aeroplane, and I imagine how the sensors are working, how everything is working in this aeroplane. Who made this? A human being made it. And still it's so complicated, and one feels amazed. And what about this human being who has every single thing which is very calculated, and the moment one small thing goes wrong, you have those very severe diseases which lead to your own death. What is cancer? We'll discuss that in neoplasia. One little mutation in the genes, and that caused the whole cancer to happen. So, subhanAllah, so see how the promoters and inhibitors will be working when they are needed. Now, the extracellular matrix, as I told you, extracellular matrix is like water for the fish. Can fish live without water? No, they love water. In the water, they are moving, they're getting their nutrition, they're having fun, isn't it? That's their life. Similarly, cells, they need extracellular matrix for their living. Because extracellular matrix control their nutrition, control their proliferation, control their differentiation, control their movement. So everything related to the cells is under control of the ECM. That is how important it is. And when it comes to repair, for the labile tissue or the stable tissue which have the ability to divide, this ECM is required to be intact so that this tissue can divide, because the growth factors will cause them to divide. But if this is damaged, then even if you have a labile or stable tissue, repair will be by scar formation. Have a look at this. This is a normal epithelium. This is its connective tissue, the extracellular matrix. Now, there is an injury. Is there damage to the extracellular matrix? No. You see, there is no damage to the ECM, so these cells will start proliferating, dividing, and will fix it, isn't it? Look at this. Now there is damage to the extracellular matrix, and there will be scar here. Repair is by scar formation. So the labile tissues, as I just told you, they are continuously dividing. They're in the bone marrow, the gut epithelium, wherever the epithelium is. Damage to the epithelium is just corrected by regeneration if the ECM is intact. Increased loss of blood cells if you have an accident and you lose a lot of blood. You just not lost the plasma, you lost the blood cells as well, isn't it? So for the plasma, you give the IV lines, you give a lot of normal saline, you're giving fluid. What about the blood cells? So basically, of course, when it's too much loss, we give them actual blood along with we have many lines there. If a person is that sick with an accident, we have cannulas in both arms. We have cannulas in the feet as well. So we want to ensure many lines. From one we are giving the blood, the other one we're giving the fluid. That's another thing. But basically, that plasma which is lost, we will replace by the fluids. But the bone marrow will then get stimulated and start producing lots and lots of, start regenerating the stem cells, and every cell line has a stem cell. Granulocytes, the neutrophils, they have their own stem cell. Erythrocytes have their own stem cell. Monocytes have their own stem cells. Every lineage has their own stem cells. They'll start dividing and producing them so that the loss of those cells can be compensated. Similarly, stable tissue, as I told you, they have the ability to regenerate, but they will only regenerate if there is a injury and if the residual tissue, the ECM, is intact. For example, 40 to 60% of the liver, more than half of the liver, you can actually give as a donation to person who needs it. And they will start dividing and make the liver back. The incision we give while cutting these transplant surgeons, they make sure that they give an incision that they damage the least to the extracellular matrix. So majority of the cells have their ECM intact, so they will divide to make the liver back. Similarly, if you have a tumor, 40 to 60% of your liver can be taken away. Liver is a very, very highly proliferative organ only when it is stimulated. There is a story, it's given in the pathology book actually, it's not a story from Google or social media. They said in the Roman era they punished a person by putting him at the top of the mountain and they tied him up. And there was an eagle who used to come and take a part of his liver from here. And next day when they come, they see that the liver is reformed again. Then it comes again. It takes out all the liver every day, but the next day the liver is back again. So what it is trying to show that the liver has a very high regenerative or proliferative ability, but it will only start proliferating when the eagle takes it out, or when we put an incision, when you give it away. Similarly, the other ones, pancreas, adrenal glands, thyroid gland, lung tissue, all these are the stable tissues, but they have the limited ability to replicate. They replicate also, but it's the liver which has the highest ability. Kidney, you remove one kidney, the other kidney becomes bigger by hypertrophy and hyperplasia. This is a stimulus to the other kidney, and then there is a proliferation of the kidney cells. Yes?
Can the proliferation happen with the reason other than an injury?
Yes, exactly. You see here, there is no injury to one kidney. We removed the other kidney. Now the stimulus is more demand. More increased demand. How does the hyperplasia occur? All the causes of hyperplasia can lead to hyperplasia in the stable tissues also. You see, what is hyperplasia? Regeneration. But today we are focusing more on repair. That's why we are talking about the injury, stimulating the cells to grow. But all the causes of hyperplasia can lead to their proliferation. Now this is the last slide. You see, this is a hepatic lobule. Can you see beautiful sheets of cells in this lobule? Remember, pneumonia, red hepatization? Hepatization look like liver. There was an alveolus, just like this lobule, isn't it? There was an alveolus. Alveolus was filled with lots of neutrophils and red blood cells. Looked like lots of hepatocytes like this, isn't it? That's why we called in pneumonia when the alveoli are filled with the neutrophils and the red blood cells, that this is red hepatization, which means that there is lots of cells giving an appearance of the liver, and because the red cells are also in there, so it's red hepatization. Now, look at this. Beautiful sheets of hepatocytes, isn't it? What are these dots that surround these hepatocytes? Any idea? Connective tissue, which is your, what's the other name? Extracellular matrix. Very good. Very nice. Now, this liver underwent an injury. Let's look at this injury first. You see the hepatocytes are dead? Their number is decreased, isn't it? But the remaining ones are there. They can regenerate. They have the ability to divide. What about the extracellular matrix? Is it intact or is it destroyed? Intact. You see, it's still in their place. Now what will happen? They will proliferate, give the liver back. See? Exactly the way it was before. Correct? Now, the same liver got another type of injury, like a chronic inflammatory injury, where you have hepatitis C causing chronic inflammation in the liver, chronic hepatitis, and every day it is causing injury to the extracellular matrix. Every day the fibrosis is occurring, scar formation, so the repair will be with scar formation. You see, hepatocytes are dead, just like here, but here the other problem is the extracellular matrix is gone. Now how will it repair? Scar formation, you see. Not the sheets of the liver will happen, and now this is going to affect the function. So remember one thing, scar formation tries to restore the structure, but sometimes their function is not retained back because of the scar formation. Example, myocardial infarction. Scar can repair the wall. Let's say there was an infarction on the anterior wall myocardial infarction. There was a big area on the anterior wall that was dyed with necrosis. Inflammatory cells come, they remove that necrotic tissue, and then because it's a permanent tissue, they are fibroblasts put in collagen, they repair the heart wall. You do the echo, you will see the scar. But now, will this wall which had this scar be as good in contraction as it was before? No. Scar has managed to heal it, managed to restore its function structure, but the function is affected. Now, if there was a big MI, this person may go into heart failure again and again because the ejection of the heart is going to be affected. So similarly for this liver, the function of the liver will be affected after this scarring. As you see, there are no hepatocytes left. Okay? Thank you very much. All the best with your exam. Remember, most of you have been very good students. You were regular in the class. I do not see any